Hi everybody. So what we're going to talk about today and the rest of this week is the idea of pathogen adhesins. So adhesins are specific molecules that help pathogens stick to their host cells. So this is incredibly important for things like establishing an infection, maintaining an infection, and as I'll show you a little bit later on, it's important for things like host range and tissue tropism. So what we mean by that are what types of host is any given pathogen capable of infecting, and what tissues is any given pathogen in any given host capable of infecting. So in other words, do you enjoy infecting the lungs? Do you enjoy infecting the bladder? Do you enjoy infecting the brain? Most, if not all of that, is dictated by the ability of your pathogen's adhesins to stick to host cells. Okay, so with that being said, I'm going to blow this guy up for a second, and we're going to look at this. So this is a figure from a paper in the journal, the CDC's journal, Emerging Infectious Diseases. This is a journal that was started in the late 90s, so this is one of its earlier papers. And what this is just a schematic that looks at what they consider to be the 4 different strategies of pathogens binding to host cells. So the first one here are using pili, or fimbriae, as adhesins. So we saw these in the very first video where we talked about components of bacterial cells. So these were the short little appendages as opposed to the big giant long flagellum that we saw. I mentioned that these are associated with twitching motility, and they have another role in genetic exchange we'll get to way later in the semester. But what many pathogens use these guys for is to actually adhere to the cells. And what you can see in this schematic diagram is that for every one of these pili that's sticking to the host cell, there's something on the host cell that they're sticking to. This is called a receptor. This is called a ligand. They have to be able to specifically interact with each other. If either one of these is not capable of having a biochemical interaction and attraction to one another and forming an actual bond, then this pathogen is not going to stick to this host cell. Okay. So fimbrial adhesions are what they consider to be the first type, and in this case what they're sticking to are host glycolipids or glycoproteins. So anything with glyco in front of it stands for something that has a lot of carbohydrates stuck to it. So these little ball and squiggle molecules here on the host cell, the ligands, these are the author's representation of either some lipids or some proteins, and then the ball at the top would be specific carbohydrate chains. Once again, these particular fimbriae have to find the right shape and size carbohydrate chains in order to bind on. So these remaining strategies are a good theme and variation on that. What you have in part B is an instance of what's called an afimbrial adhesin. So this is a receptor molecule that's directly on the surface of the pathogen. So it's not extended out at the tip, it's right directly on the surface. In the same instance, the host cell, the ligand in question, works the same way. It's protein or lipid, and it's typically glycosylated. In this case, the pathogen sticks right straight onto the cell membrane as opposed to being kind of up here somewhere. In this case down here, where you have pathogen receptors interacting with host integrins. So integrins on host cells are molecules that actually help the host extracellular matrix grab on to the host cell membrane, and then they actually link it to the actin and myosin layers underneath. In case anybody needs a refresher from cell biology, actin and myosin layers underneath the cell are what help this cell, this host cell, retain its shape and actually move and rearrange its cytoskeleton. So when you have pathogen receptors that are interacting with integrins, what you have, and by the way, these types of receptors are called intimins, as you can see right here. These guys are now able to shift and move the actin cytoskeleton of the host cell, and they're able to reshape the host cell as they see fit. Finally, the last type of receptor, adhesin that these guys have demonstrated would be a secreted adhesin, one that the actual pathogen secretes out of its surface. It's then integrated into the host cell, modified by the host cell, put back out on the surface, and then the pathogen itself has another mechanism of binding it. The common theme in all of these is that you have to have the right pathogen receptor and the right host cell ligand, that these are very much a lock and key type of interaction. So to take another view of receptors and ligands, this is actually a figure that I borrowed from a journal article, this one right here. It was published last April. This looks at a new adhesin called FDEC from E. coli that is actually just first described in this paper. And what they did was they actually made protein structural diagrams of this adhesin. They looked at different regions encoding it, and each different region was responsible for different interactions with the host cell ligand. And what they're doing in this particular picture is that they're showing you the protein structure of FDEC, their new adhesin, and then they're showing you the similarities and differences between the adhesin intimin in a species called Yersinia pseudotuberculosis, and another, I said something backwards. Please blame Dylan from section 1 because he appeared in my doorway and distracted me. Ha ha ha. The point of this particularly panel D in this figure is to look at the adhesin FDEC, to look at the actual crystal structure and shape of the protein, and to compare it to two other adhesins, intimin from E. coli and invasin from Yersinia pseudotuberculosis. Both of these adhesins are actually mentioned in that previous figure on slide 1 from the Emerging Infectious Diseases paper. The point here is to show that these guys may have very similar basic shapes, but they have difference in sizes, difference in molecular weight, difference in regions, and so these differences are going to give them an equivocal ability to adhere to cells in the same way. In other words, different shape of the adhesin gives you a different predilection for binding different host cell ligands. Okay, so with this last bit, the thing that I want to communicate to you all is that a human cell is not simply a human cell. We have different types of cells, right? We have lung fibroblasts, we have tracheal epithelial cells, we have neurons, we have hepatocytes, we have macrophage, etc., etc. We have a ton of different types of cells. Not every pathogen binds every type of cell. The reason for that goes back to that adhesin-ligand interaction. In addition to that, we also have the idea that not every tracheal epithelial cell is interchangeable. So if you stop and think for a moment about rabies virus and the fact that rabies virus infects neurons from many types of mammalian species, right? So you have humans and raccoons and foxes and dogs and a whole range of other things. Yet it does not infect neurons of a chicken or a cardinal or an eagle or a salamander or a crocodile. The point there is that rabies virus has an adhesin, an attachment mechanism that sticks to a ligand that's fairly widespread on mammalian neurons, not universal, but fairly widespread, and that whatever that ligand is, it's not maintained and it's not conserved across all animal species, because you don't see rabies viral infections in birds and amphibians, reptiles, or fish. So one of the things I want to try to impress upon you is how that host ligand and pathogen receptor interaction develops. Where exactly does the specificity come from? And in many cases, it has to do with glycosylation patterns on the host ligand. So what I mean by that is what carbohydrates are modifying that host ligand, and how are they linked to one another. So that doesn't sound very complicated, or conversely, perhaps it sounds extremely complicated, but it's not too bad. It's pretty intuitive, actually. So what I want to do is just describe a couple of common carbohydrate modifications that one might see on this, either on the surface or in the extracellular matrix of mammalian cells. So the first one would be something called hyaluronan. Another one could be galactose. We'll call them HA, gal. Another one would be N-acetylglucosamine. We'll call that GLICNAC. In case you couldn't tell, that is not my abbreviation. We've also got sialic acid. I'm going to erase that and make it BSA. And another one could be something called heparin. I'm just going to call that H. So these guys have very big, complex molecular structures, but we're going to think of them in terms of cardinal shapes to make things easier. Okay, so if we think of hyaluronan as being shaped like a triangle, and each point in this shape is a carbon molecule. So this is carbon 1, this is 2, and this is 3. Let's think about galactose for a minute, and we're going to make galactose be shaped like a pentagon. Here, it's carbon 1, 2, 3, 4, and 5. Let's make GLICNAC be shaped like a hexagon. And here we have carbon 1, 2, 3, 4, 5, and 6. Sialic acid we're going to make into a star. Carbon 1, 2, 3, 4, 5. And heparin, we're going to make that be a big giant rectangle. So here we have carbon 1, 2, 3, and 4. I have bad handwriting with an actual pencil, so now you get to deal with my finger. It's not good. So let's think for a moment, or let's pretend for a moment, we have a host cell protein, and it has a hyaluronan molecule, and that is linked to a sialic acid molecule. That is linked to a heparin molecule, and that is linked to a GLICNAC molecule. All right. If we have them linked like this, okay, so here's our hyaluronan, here are our carbon numbers. Here's our bond between the carbohydrates, or that would be called a glycosidic linkage. Here's our sialic acid. And let's make this be carbon 3, 4, 5, 1, and 2. And then we have this carbon 1 being linked over to our heparin. And let's make this be carbon 1, 2, 3, and 4. Excuse me. And then we're linked at this point to a GLICNAC. Now let's make that come from carbon 3 and bond over here to our let me draw that a little better. Have that be bonded to carbon 6 over here. So here's 6, 1, 2, 3, 4, and 5. Okay, that's great. So if we were to look at our chain of carbohydrates, we might think, all right, that's what it's just going to look like every time you do this, but it's not true at all. And in fact, you can come up with something extremely different. I'm going to just take a moment and shrink this a little bit so I give myself more space. If we had our hyaluronan, and then all of a sudden we started doing things a little differently and said, all right, here's carbon 1, here's 2, and here's 3. If we now, instead of bonding carbon 2 on the hyaluronan to carbon 3 on the sialic acid, and let's say we bond carbon 1 to carbon 4, for example, on the sialic acid, so here's 4, 5, 1, 2, and 3, that's great. What if we then take carbon 3 on the sialic acid instead of carbon 1, as we have over here, and we bond that to our heparin, except we do it to carbon 4 instead of carbon 1. Now all of a sudden what we've got is something that looks like this. 1, 2, 3, 4. So you can kind of see that our heparin, instead of being upright like this, is now kind of flipped upside down and sideways, and now it looks like this. And if we then bond our glycnac from carbon 1 here to carbon 2 over here, this is 1, 2, 3, 4, this would be 5, except I don't have space to draw it, and 6, we can now see that even though we have the same 4 carbohydrates bonded to each other, if you start bonding them to different carbons, you're really changing the shape of this glycosidic modification that's on the surface of a host cell. So if you think back to that first image we saw when we had these nice kind of, the ligand looks like this on the host cell, and the receptor looks like this, and you have this kind of very nice lock and key looking situation. If your lock normally looks like this, and then suddenly by changing what carbons are bound to which, your lock now looks like this, your key isn't going to fit anymore. So that's actually pretty substantial change. So if I can if I can take these two guys, what's going on? If I can take these two guys, move them at least out of the way if I can't shrink them, and draw you another little point. The way that you write these linkages out is different, as you could imagine. So in this first case, we would write HA, it's linked to sialic acid, linked to heparin, linked to glycnac. In this first case, what you do is you describe the linkage by what carbon number is bonded to which one. Here you have carbon 2 on the hyaluronan, and it's bound to carbon 3 on the sialic acid. In this case, between sialic acid and heparin, you have carbon 1 on sialic acid bound to carbon 1 on heparin, so you have a 1-1. Here you have carbon 3 of heparin bound to carbon 6 of glycnac, so you have a 3-6 linkage. Down here, you'd write this, same carbohydrates, but you're going to write the actual numeric designations differently. So you write hyaluronan, sialic acid, heparin, glycnac, and here you're going to write, we're starting from carbon 1 on hyaluronan, and we're bound to carbon 4 on sialic acid, right here. So that's here and here. On sialic acid to heparin, this linkage looks at carbon 3 to carbon 4. So here you're going to write 3, 4. And then heparin to glycnac, you're doing carbon 1 to carbon 2. So you're 1, 2. Okay, so if you looked at a series of carbohydrates and looked at a series of strange numbers in between them, the differences between these numbers is going to give you an idea of what carbon is bonded to which one on each carbohydrate, and then geometrically what's going to start happening is the more times you see these being different, you can start to envision that you're going to have a slightly different shape. To complicate things further, it's not just which number carbon is important in terms of how glycosidic linkages work, it's also what type of linkage, as in, is it symmetric or is it asymmetric? So oftentimes carbohydrates have R groups on them, or external structures that give some kind of stereochemistry to them. So if we start looking at hyaluron-N, sialic acid, heparin, and glycnac from the standpoint of their R groups, so if we say, in this case, we put a little modification on carbon 3 of hyaluron-N, and we put a modification on carbon 5 for sialic acid. Let's put a little asterisk right here. If we put one on carbon 3 of heparin, put a diamond here, and if we put one on carbon 3 of glycnac, let's put an X here. That's something I should be able to actually draw effectively. And then we start looking at their linkages and how the shapes change with regard to how these modifications wind up in terms of their symmetry or asymmetry. Now we have an even greater layer of texture to add to this. So if we shrink this down for a second again, so we have enough space to draw, let's try the first version that we drew again a second ago. So if we take hyaluron-N, we take carbons 1, 2, and 3, and now we add our little modification to carbon 3 here. Our first linkage was a 2,3 to sialic acid. So here's our sialic acid molecule. Here's carbon 1, 2, 3, 4, and 5. If we put our modification on carbon 5 up here, and then we linked, this is not looking real good. This is a star with too many points on it. Hold on a second. Sorry. If we then linked by carbon 1 to heparin, we draw our big rectangle up here, and we're linked to carbon 1. Here's 2, 3, and 4. We put our little diamond modification on carbon 3, as we said, and then we linked carbon 3 over here to carbon 6 of glycnac. 1, 2, 3, 4, 5. And we put our modification over here. Sorry, that's supposed to be just an X. Oh, and here I said I could actually draw X's. Apparently I lied. Sorry. What I mean by symmetry and asymmetry has to do with the idea of alpha and beta glycosidic linkages. So, as a reminder, and again, I've put an introductory biology lecture about glycosidic linkages up on YouTube if you'd like to review it. When you have modifications on opposite sides of a glycosidic bond, like in this case, this is asymmetric, this is a beta linkage. When you have modifications on the same side of a glycosidic bond, so right here's the bond, this one and this one are on the same side, that's an alpha linkage. And so now, here's your linkage, here are your modifications. Ask yourself quickly what type of linkage that is. Please pause, because I'm going to write it in a second. Okay, hopefully you wrote alpha. So when you're truly defining what this linkage is called between this heparin and this sialic acid, the way you'd actually write this out remembering our numbers from the previous slide, is you would write this out as a beta 2,3 linkage. That's what that bond is called. So this one right here, again, please feel free to pause and see if you can fill in the blank here, but this one is an alpha 1,1 because we have our linkages being on the same side of the bond. That's where you get the alpha. The first one comes from the carbon number on sialic acid. The second one comes from the carbon number on the heparin. And so finally, again, pause here, see if you can come up with the answer. After pausing, hopefully you're thinking to yourself that this is an alpha 3,6 linkage. So if we look back to our, I'm going to draw a big line and then shrink this even more. If we think back to our other glycosidic linkage patterns that we drew on the previous slide, where we started linking different carbons. So if we start with our heparin again, and we put carbon 1 here, 2 here, and 3 here, and we put our little modification on the, I'm sorry, this is hyaluronan, not heparin, and then we link that to carbon 4 on our sialic acid molecule. So if this is 4, this is 5, this is 1, 2, and 3, and then we put our asterisk on carbon 5, and then we link from carbon 3 down here to our heparin molecule, and we link that to carbon 4, 3, 2, and 1, and carbon 3 is where we have our little diamond modification. And then finally we linked carbon 1 on the heparin to carbon 2 on the glycnac molecule. That's the worst hexagon ever, sorry. Here we go. So this is now carbon 1, 2, 3, 4, 5, and 6. And if we had our modification on carbon 3, what we would then do is see, all right, now I've got different glycosidic linkages because I've got different linked carbons, but now since my geometry's changed a bit in this, this new ligand glycosylation pattern, I now presumably have a different mix of alpha and beta linkages. So if we look at this first one, we have this modification and this modification on the same side of the linkage. So this is an alpha 1,4 linkage. Right here we have this modification here. Here's our linkage. Here's the modification on the heparin. So in this case we now have a beta 3,4 linkage. And then in the last case, here's our bond. Here are our modifications. So we have an alpha 1,2 linkage. So hopefully what you can see with all of this alpha beta number geometry R group business is that not only does the order in which glycosidic linkages stack up matter. So if we suddenly change this order of things, if we said, okay, the first sugar that's bound to the cellular protein is glycnac, then you have 2 sialic acids, and then you have a hyaluronan, and then you have 4 heparins, that's really going to change the geometry of the host cell ligand that the pathogen now has to bind to. But if you start flipping around what carbon number is bound to whom, you really start to change the shape. And then when you consider that most of these carbohydrates have modifications on them, and we now have to think, well, are these asymmetric linkages, or are these symmetric, or are they asymmetric linkages? There really is an incredible amount of diversity that you can get in terms of host cell ligands. And so pathogen adhesins can either be very promiscuous in their abilities to bind these, meaning they can bind all sorts of different glycosidic linkages, or they can be extremely specific. If something is extremely specific and binds only these 4 carbohydrates in this order, in this shape, with these modifications, then it's going to be a pathogen that's extremely specific for the tissue and host that has this ligand. If it's less fussy, it's going to be able to have a broader host range and a broader tissue tropism. In other words, it's going to be able to bind multiple body sites. And since a pathogen binding on to host cells is paramount in the infectious process, these types of linkages right here are what really dictate a pathogen's host range and target tissues. One last point, I promised I would do this because he was called out for asking a question that allowed him to do legit science and use the appropriate plates in the plate reader. So, sorry, Dylan. Have a good day, everybody.